Hey guys, it's Miss Meckling again. We're going to do this set of notes, and I'm going to try to adjust as much as I can as we go. And your job is to fill this in as we go and then submit it. This first page is all about DNA replication, and we'll watch a video on how this works. But it's pretty straightforward, okay? So up here it says the building blocks of the DNA molecule are nucleotides, which consist of a phosphate, a deoxyribose sugar, and a nitrogenous base. In the diagram, you are to label all three substances on the nucleotide. The letters represent the four different nitrogenous bases, are shown in the nucleotides at the right. Place the name of the base next to the letter symbol in the appropriate space. Okay, so this diagram asks you to label, number one, the phosphate, then the deoxyribose sugar, and the nitrogenous base. Okay, so yes, just like the video we watched last week, the phosphates are the circles. Okay, and if you look, there are 1, 2, 3, 4, 5 at the top, and 1, 2, 3, 4, 5 at the bottom. So there are a total of 10 phosphates. And remember, phosphates are phosphorus, so P on the periodic table, with 3 oxygens surrounding them. So it's actually 4 different elements in them. Then these kind of like pentagon, not pentagram. Jeez, it's Sunday, just bear with me. All right, the pentagons are the deoxyribose sugars, okay? So OSE ending is a carbohydrate. Remember, it has carbon, hydrogen, and oxygen only. Therefore, all of these in the middle, the C's, the G's, the A's, the T's, they are all the nitrogenous bases. Okay, nitrogenous bases. Okay, so you need to label those. There are, if you look, 1, 2, 3, 4, and this doesn't count, 5, 6, 7, and 8, and then this extra phosphate. So there are 8 total nucleotides. Okay, and the nitrogenous bases, remember, A stands for adenine. Okay, T stands for thymine. G stands for guanine. And C stands for cytosine. Okay, remember, this is DNA here. So A goes with T, apples in the tree, and cars in the garage. So that's why you always see an A here and a T here and vice versa. All right, so that's the structure of DNA. So again, the spellings are down here later. The DNA molecule has a double helix shape, means it's double-sided, and it's a helix. It's a ladder that's twisted, as you can see in this image. 2 strands of DNA are coiled around each other and attached by bonds. Remember, we said those bonds were hydrogen bonds. It was an attraction between a positive hydrogen and a negative hydrogen between the nitrogenous bases of each chain. Adenine always bonds with thymine, and there's your spelling again. You need to know how to spell them. And cytosine always bonds with guanine. Remember, apples in the tree, cars in the garage. All right, so now these go to these diagrams down here. So I'll move it up a little bit. Okay, and I'll try to zoom in a little bit. So this first one right here says, in the illustration at the left, label a phosphate. So that's the first thing you need to do. Label a deoxyribose sugar. You just have to label one of them, okay? And then fill in the symbols for each base depending on its complementary base in the opposite strand. So that's what we're doing on this diagram here. So again, the phosphates, and you got to put an arrow when you do these notes and turn it in, okay? So the phosphates are the circles. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9 on that side, and 1, 2, 3, 4, 5, 6, 7, 8, 9 on the other side. So you have 18 different choices. Okay, just label one. Doesn't have to be the same one I labeled. Okay, so the phosphates. Then the sugars, again, are that weird shaped pentagon. Okay, so the deoxyribose sugar, okay, are the shaded in, kind of looking like arrow things. Okay, and then we have to fill in this chart according to the complement. So here on this side, we have a T, so you're going to label this side an A. C goes with G. Now we've got this unknown, but it's the same shape as here. So T and A. Here we've got, it looks like a G, so we're going to mark a C. And we'll do that again, a C again. And then A and T, C and G, and the other one we can't really see. Okay, so that is what we're doing on that side. This side, however, it says the diagram at the right below, at the right, shows the replication of DNA. So this is the first part we're going to talk about in our genetics unit. So it says the diagram below shows the replication of DNA. Fill in the symbol of each base. Label, here, I'll make this a little bit bigger, okay? Fill in the symbol of each base and label the original strand. So you gotta label the original strand, the new strand, and the free-floating nucleotides, okay? So we start with, let me use my purple pen over here. Okay, we're starting at the top. So right here, if you follow, let me see if my pen works. Well, all right, so this purple line right here and this purple line right here. Okay, so that is going to be your original strand. Original strand. All right, that's your original strand of DNA. It starts at the top, it's still together, and it's unzipped. Okay, we'll talk about what does the unzipping later on. The new strand is going to be built from the bottom. So let me get a highlighter. Just use the pink one. Okay, so this right here that I'm highlighting pink is where it's building the new strand. So starting at the bottom, and I'll use orange, and I'm going to circle these three right here. 1, 2, 3. Those are what we're going to call, oh, don't forget to label it, new strand. And you can do it twice. Okay, so this is the new strand. You see that? Let me make it bigger. All right. I don't know how to get this in the image. Here we go. So this pink right here is the new strand. All right, and then these three, 1, 2, and 3, are not attached to anything. So those are free floating, and you see they have all three pieces, free floating nucleotides. Okay, so nucleotides. We got 3 of them. All right, so the last thing it asks you to do is to fill in the symbol for each, on each. So here, if you look at the symbol from over here, you can tell the ones with the little arrows kind of look like A's, and then this is a T. All right, so the ones that look kind of like a Pac-Man, like these free-floating ones, they're all T's, okay? So this is a T, this is a T, this is a T. All right, and then the other ones that look like full circles are G's. So then that would be a C, kind of looks like an inward part. The one that we said that was arrow was A and T, and this is the flip-flop of that. Okay, so T and A. And over here, looks like we have the full circle again, which is G, G, and can you see that? G and C. I think that's all we can see. All right, so that's how you label that diagram. Then at the bottom, it's kind of a fill in the blank. So this might be some new information. So it says, don't mind my nails. The structure of DNA was determined by 2 scientists, James Watson and Francis Crick. Francis. Let me spell it right. Oops, it's a C-I-S. Crick. Okay? And they determine the shape of the molecule to be a double helix. Okay, so this is how we get one copy of DNA to make two. So it unzips itself, and then we use the original strand as what's called a template. Okay, templates are used, especially in art class, to repeat a pattern. So if you were doing an artwork and you wanted to use the same pattern of square, rectangular, triangle, and you would cut it out on a, say, cardboard, and then you trace it over and over again. That's what a template is. So this original strand becomes the template, and then they make the new strand from the old template. Okay, after replication, you have two identical molecules of DNA are produced. So two identical molecules from the original one are reproduced. And a gene is a sequence of nucleotides on the DNA molecule. So it's just a small segment, but it's not the whole strand like we talked about last week in our video. Okay, so it's a sequence of nucleotides in a DNA strand. Okay, so that's page 1 of the notes. Now page 2 is new stuff. So new. Okay, we talked about last week the 3 different types of RNA and the differences between RNA and DNA. Obviously the sugar, ribose versus deoxyribose. RNA is single-stranded and DNA is double-stranded. And then the other thing is that DNA can leave the nucleus. Okay, so remember I used the example of grandma's recipe. I think I used chocolate chip cookies and maybe macaroni and cheese. So grandma doesn't let you take that recipe out of the house. So you want to make it at your house, you've got to write it down, make a copy of it, or you've got to take a picture of it. Okay, so that's kind of what's happening here. We're going to take the strand of DNA and make messenger RNA out of it in a process called transcription. Okay, if you break it down, scription or script is something written. So if you read a script of a play, it's the written form of the play. So what we're going to do is we're going to make a copy of the DNA. So here it says, fill in the blanks below. And on the illustration of transcription, which is DNA, taking DNA and making mRNA from it, label the DNA. Okay, you got the newly formed DNA, we'll call that number 1. Or I'm sorry, label the DNA is 1, the newly formed is 2, and the completed strand of messenger RNA is 3. And the last one, number 4, we'll just label it number 4 because these lines are kind of small. We'll just call them 1, 2, 3, and 4. So 1, 2, 3, 4. We will label them with numbers because the lines are really, really short. So let me give you these notes, and then we'll do this, and then we'll continue at the bottom. All right, so it says messenger RNA, or M, lowercase m. Remember, this is a little m. Messenger RNA carries the instructions to make a particular protein. Okay, so remember, DNA are the instructions to make proteins, but we got to take that recipe to make the right protein to the place where proteins are made, the ribosome. Okay, so we got to take that message or the instructions from the DNA in the nucleus to the ribosomes, which are either, remember, free floating in the cytoplasm or attached to the endoplasmic reticulum, calling it the rough ER. Okay, the process of producing messenger RNA from instructions in the DNA is called transcription. Right here, up here, transcription. Okay, here's the word again. During transcription, the DNA molecule unwinds and separates. So that's step 1. Exposing the nitrogenous bases. The free-floating RNA nucleotides, okay, and they have to be RNA, where ribose is the sugar. The free-floating nucleotides pair up with the exposed bases. there is no thymine, so there's no T. Sorry, excuse me. There's no thymine, T, in RNA. U, which is uracil, replaces the T, and that will pair with A, adenine, instead. So remember, there's no T, it's only U. RNA contains the sugar ribose. Remember, OSE ending is a carbohydrate, instead of deoxyribose, which is the sugar that's in DNA. The messenger RNA molecule is completed by the formation of bonds, so we get those hydrogen bonds again, between the RNA nucleotides okay? So we're going to create bonds again between those nucleotides, those bonds that we separated to unzip the DNA. And then separates from the DNA, and it's released. The messenger RNA molecule is a single strand, and it's single, unlike DNA, so that it can actually fit through those nuclear pores. If it's kind of like taking a ladder, you can't fit a ladder sometimes through a window, but we cut that ladder in half, we might be able to get it out. That's exactly what happens in the nucleus, okay? So over here we have the diagram. Let me see if I can pull this to the side. There we go. All right, so now we got to label the DNA, the newly formed messenger RNA, the complete strand of messenger RNA, and the free-floating nucleotide. So the one that's easiest are these guys out here. So number 4, see how this one, and this one are free-floating? They are not attached to anything. So that is your free-floating nucleotide. Okay, we start at the top, and if you look here, that is your original strand of DNA. So if I were to take my highlighter, and I started at the top, and I highlighted it all the way down, okay, this is part of the original, okay? So the original DNA, number 1, is right here. Then we have 2 more choices. The newly forming messenger RNA, okay, let me scroll that down. The newly formed messenger RNA and the completed strand. Well, down here, I'm gonna make this orange, okay? It's not attached to anything. So that has got to be the completed strand, okay? So that's number 3. But this is also RNA right here, so I'm going to use the same orange. But as you see, it's still attached to the DNA. So that would be number 2, the newly formed RNA. So we unzip the DNA, the free-floating nucleotides, where this sugar is now ribose, will come in and pair up. Remember, apples not in the tree anymore, they're under the tree, and cars still in the garage. Okay? All right, so that's called transcription. It's pretty easy. It's the same way we do DNA replication. Instead, it's RNA, and instead of thymine, we have uracil. Okay? So now here at the bottom, you kind of got a hint of this, and this is what you're going to do when we do our assignment on Tuesday. So remember, Tuesday, you got to come to class to get the assignment. Okay. So Tuesday, we are going to do a scavenger hunt. So I will help you start it, and the last thing you have to do is you'll have to find the messenger RNA, and then you'll have to translate it. Okay, so this is what we're translating it back into the right amino acid. So it says down here at the bottom, each combination of 3, this is the keyword 3 nitrogen bases on the messenger RNA strand. So it would be right here. Say 1, 2, and 3. If you have 3 nitrogen bases right there, they get read, and they get read by the ribosome. So it's kind of like sticking, like if you ever remember going to Chuck E. Cheese and sticking those little tickets in the machine and eats it up and counts them. That's kind of what the ribosome does. It eats up the little ticket of messenger RNA, and it reads every 3. And every 3 is going to code for a specific amino acid. Okay, remember, amino acids are the building blocks of proteins. The table below shows the messenger RNA codons for each amino acid. Use the table to answer the following questions. So the key thing is the first base you read along this side, just kind of like your multiplication table. The second base is here, and if it was multiplication, right, you would where they intersected, and then we've got a third base over here. So like this one, if my first base was U, my second base was G, it would put me in this box, and then I would use this last one, say it was G, it would give me tryptophan, which is what's in turkey, makes you sleepy. Okay, so look at that. Oh, the codon for tryptophan. So tryptophan is right here. So U, G, and it's the second row, G. So UGG is the answer for the codon for tryptophan. Okay, however, sometimes there are, because there's 20 essential amino acids, there are more than one if you count up how many different. So if we have 1, 2, 3, 4 in each box, and we've got 4, we've got 16 in this row, times 4, there's 64 combinations, right? But we only have 20 essential amino acids. So there's ones that code for a start and a stop. So it tells them to start reading and then stop reading. And then there are some that are duplicates. So this question 2 says, for leucine, there are how many different codons? And if you look, leucine is 1 right here, 2 right here, and then we got 1, 2, 3, 4. So we actually have 6 codons for leucine. And that's kind of nature's way of correcting mistakes. So if you look, this would be C, U, and A, C, U, and G, C, U, and U, C, U, and C. So if that last codon was anything else, it would still get the same thing, okay? The codon G, so we're going to go right here. A, so it puts us right here in this box. And U, which is the second to last one, is aspartic acid. And aspartic acid is what's in asparagus. Again, spelled very similar. I don't want to misspell it. Okay, aspartic acid. All right. And the last one, it says, in a stop codon, if the second base is G, so it's got to be in this column, what are the first 2? So the first one would be U, and the second one would be A, U and A, okay? So that's page 2 of our notes. So this, once you get more practice with it, it's fairly simple and straightforward. I would never ask you to memorize this because just like the periodic table, you can get access to it. It's in the textbook, it's online, and I will give you a copy when we do the assignment on Tuesday and Wednesday, okay? All right, so now page 3 is just kind of a review of everything. So let's go through this and then we're done. The last page of this notes is the kind of the codon chart that you need for the scavenger hunt. So I just put this here as a reference, okay? And it's kind of funny. It says, isn't genetic engineering amazing? 2 years ago, who would have ever imagined such thing as a turkipede? So he has 1, 2, 3, 4, 5 legs, 6 legs on one side and many on the other. So wouldn't that cause you not to fight over the turkey leg at Thanksgiving? Anyways, kind of funny. Anyway, so that page you can just kind of set aside and make sure you have access to it for the assignment on Tuesday and Wednesday. All right, so I'm going to try to make this bigger so you can see it. So question 1 says, transcribe means to copy, okay? So to copy. So you're transcribing your grandma's recipe when you write it down or she writes it down for you. Translation is putting something in a new form, okay? So just like you do in Spanish and French and Japanese or Italian class, you transform it into a new form. Okay, going from DNA to messenger RNA is called transcription. Transcription. Because the messenger RNA is a copy of the DNA. Going from messenger RNA to a protein is now called translation. So you're translating it, or translation, sorry. Translating it. In DNA, A pairs with T, so apples in the tree, and C pairs with G, car in the garage. In RNA, however, A pairs with U, apples under the tree, and C still pairs with G, cars in the garage. Proteins are made up of amino acids, And AUG, if you look back at the chart, I don't know if it was on that last chart. AUG, if we look at this chart again, A, U, and G right here. Code for this protein called methionine. And methionine is actually called a start proton, or protein. So your anything that's, say, on the RNA up here, it won't read until there's actually a methionine there, and then it starts reading. Okay, so and then it'll read until you get to a stop codon. Okay, so AUG, which is what you're going to need to know for our scavenger hunt, is what you're going to be looking for this codon as the start codon. Okay, and then UAG is just one of 3 stop codons. Okay, so there's 3 of them. I don't know why. Okay, there's 3 total. If you look back here, UAA, UAG, and again, this one, UGA. Okay, so up here it had UAG, UA, and G. So there's also UAA. UAA is another one. And the last one is UGA. UGA is another one. Okay, so there's actually 3 stop codons. All right, messenger RNA is made in the nucleus, but it doesn't stay in the nucleus. But it gets translated, and we can call it two different things. It gets translated in the cytoplasm, but more specifically, it also gets translated, more importantly, at the ribosome. Okay. Number 9, 10, 11, and 12 are something I'm going to explain in a second. But number 9, the last review one, it says the ribosomes are the protein-making machinery. So ribosomes. So that's where proteins are made. Now, down here, it says new things, new thoughts. And if we were in the classroom, I could show you like a small video on this, and maybe I'll try to pull it up and post it with this. It says in a firefly's tail, thousands of different enzymes called luciferase. Lucifer, the devil, right? Luciferase enzymes are produced at once. This enzyme combines with other chemicals for a reaction that produces light and causes the glowing that we know of fireflies or lightning bugs or whatever you call them. But this is what happens. It translates it into the right enzyme all at once, and then it gets to actually glow. And remember, it's just the males that are doing it, and they're trying to attract a mate. So whenever you're catching a firefly in the summertime, you're keeping that firefly from mating. So don't block them, right? All right. Number 11 says the GFP, which stands for green fluorescence. I'm going to spell it wrong. Nope, see, I did. FLOUR. So kind of like flour, let you cook with. Scent, fluorescent protein. Okay, so the green fluorescent protein is a gene actually produced from a jellyfish and can be transferred into any organism. In fact, there was a cat, I want to say, on one of the National Geographics that they put it in. But it can also be transferred into a pig because genetic code is universal. Okay, this is also how we're able to take like body parts, like, for example, a heart valve from a pig and transfer it into a human. We are all made of those C's, G's, A's, T's, and U's. So it's a universal code, okay? It's a universal code that makes all living things. And GFP is very important because it can be used in many organisms for research. So we can actually figure out if that specific gene along that DNA is being used or not by cutting that out, that section out, inserting this section for this GFP to be produced, and then seeing if it actually glows under a black light. Okay, So it's kind of a really cool thing. And anytime you have the genome or a section of the genome of someone else's DNA inserted into another organism, it is called transgenic. Okay, transgenic. And transgenic organisms are organisms that are genetically engineered by inserting the gene, or sometimes it can be an organ, okay, from another organism. And remember, a codon, last thing is, the codon is a combination of 3 nucleotides that code for an amino acid. And the last thing we got for today, super quick, this goes down, so it goes down like this, okay? So we've got DNA transfers and transcribes into messenger RNA. Messenger RNA is translated into the amino acids that build the protein, okay? So transcription, where does it take place? It takes place in the nucleus. What is used? DNA, and it unzips and uses free-floating RNA nucleotides, okay, that code in the same sequence. And the end product is the mRNA. Okay, so that's the simplest way I can explain to you what transcription is. Okay, translation, where it takes place, is in the cytoplasm, or more importantly, the ribosomes. Okay, and what is used? The messenger RNA is used, and then it's read in the ribosome, and the tRNA will come in, and we'll watch a video on this next week. The tRNA will come in and has what we call an anticodon on it, will actually drop off the right amino acid in the right sequence based on the recipe on the mRNA, and you end up making the protein. And we'll watch a video on that. Okay, protein, let me spell it right, T-E-I-N. Sorry about that, guys. Okay, so for example, if I have CGG, and this was on the mRNA, okay, the tRNA, which we saw in the video, kind of looks like this, right? And then it has these 3 codons at the bottom. This is called the anticodon. Okay, and the anticodon would have to pair specifically with that codon in the messenger RNA. So if we have C there, we'd have G, C, C. And then it would fit into there, and it would drop off the right amino acid. And we'll see that in a video in class next week. All right, like I said, this last page here has the codons for the messenger RNA, which will translate into the right letter for you to figure out what the sentence says. And I'll go through that on Tuesday and Wednesday. Thanks, guys. Have a great night.